Today we have Dr. Dmitry Dukhovskoy, who is a research scientist at Florida State University and a member of the POS panel as well, who is going to talk to us about freshwater in the subpolar North Atlantic. So thank you, Dmitry, and if you wouldn't mind going ahead and starting.
Yeah, thank you, Aaron. Thank you, Jenny, and good afternoon, everybody. Today I'm going to present my research on Greenland freshwater in the subpolar North Atlantic seas. This study was funded by the ONR and DOE. The major goal of the ONR funding was to develop the modeling system for the high latitudes, as well as to study the freshwater fluxes in the Arctic subarctic domains, whether the DOE project was mostly focused on studying the heat fluxes onto Greenland shelf from the North Atlantic. And many ideas that I will be presenting here today, they're still arguable, they're still hypothesized, so they're not probably certain. And these ideas came to discussion during our famous workshops. Some of you probably participated. So I would acknowledge Andrei Prushtinsky, Igor Ishaev, Jonathan Bamber, and Paul Myers for taking part in this discussion as well. So this is our domain of the study. The subpolar North Atlantic is contored here. This is the main focus of my talk today. And the diagram also shows schematically the mesoscale circulation around Greenland and in the subpolar North Atlantic. And as you know, Greenland is one of the important, not the largest one, but important source of fresh water in the North Atlantic. If we compare in terms of freshwater volume, it is comparable. It's about 30% of freshwater flux through Fram Strait. Although we need to keep in mind that these freshwater estimates derived from oceanic fluxes are so-called freshwater fractions. So I'm not going to talk about this. Just keep in mind that these fluxes are maybe not 100% comparable to the pure fresh water that I will be talking about today. And if you're interested, you can read more in this wonderful paper by Schauer and Loh, recently published in JPO. So I'm going to talk about the Greenland freshwater anomaly, because when we talk about the impact of increased Greenland melt on a thermohaline process in the North Atlantic, we mostly mean the anomaly. Because if we go back in time, and I'm using data from Jonathan Bamber, if we go back in time, we see that there was very little variation in the annual Greenland freshwater discharge from year to year. The overall mean is shown with the black line, and only in recent 3 decades we observed the increased freshwater discharge from Greenland, which is shown over here. So the definition of the Greenland freshwater anomaly is a time integral of this perturbation flux from time t0, which is year 93 or 94, forward in time. If we integrate this anomaly from 93 through 2016, the Greenland freshwater anomaly is about 5,000 cubic kilometers, which is a substantial amount of freshwater dumped into the North Atlantic. It is comparable to the amount of freshwater accumulated in the Beaufort since 2003, and it is about half of the freshwater volume advected to the North Atlantic with the GSA, Great Salinity Anomaly of the 1970s. So given this fact, there were several hypotheses put forward about possible impact of Greenland freshwater on thermohaline convection, thermohaline processes, etc. in the North Atlantic. And we also hypothesized back in 2015, I guess, that this surplus Greenland freshwater could disrupt the decadal variability observed in the 20th century between the Arctic Ocean and the subpolar North Atlantic. So the question is it really so or not? When we look in the observations, we couldn't find any obvious direct evidence of Greenland freshwater presence in the subpolar North Atlantic seas. These are the time series of salinity anomalies in several locations at the SPNA, the subpolar North Atlantic, and you can see that they do not correlate very well with the Greenland. Actually, they don't correlate at all with the Greenland freshwater flux anomaly, which is shown at the top of the panel. The recent decrease of salinity is the recent freshman in the North Atlantic, freshman of 2010, probably we can call it such way, but again, we cannot directly relate this freshman with the Greenland freshwater flux anomaly. So the question is, why is the impact of the Greenland freshwater anomaly, although kind of comparable to the volume of the GSA, it's not so evident. And another question, how long does the Greenland freshwater stay in the SPNA? Because maybe all this freshwater is quickly removed from the subpolar North Atlantic. That's why we don't see any impact. In other terms, we're interested in the time scales of the Greenland freshwater anomaly in the subpolar North Atlantic. And of course, the related question is, what is the pathway of the Greenland freshwater anomaly in the North Atlantic? So to study this question, I use numerical methods, and arguably, I think numerical methods is the only way we can study the Greenland freshwater anomaly in the North Atlantic. The reason why I say this, because it's very difficult to track salinity anomaly in the real ocean. The signal quickly dissipates in time and in space. Also, it can be masked by other processes such as river runoff, precipitation, or local ice melt. Of course, we can use tracers to track meltwater, and this has been done in situ observations. But to my knowledge, and correct me if I'm wrong, we cannot really evaluate, quantify the presence of Greenland freshwater in the subpolar North Atlantic. And here is the reference of papers where you can see more details about the model, about the analysis and validation of the models. As I said, I'm using the model. This is a coupled modeling system, HICAM and CIS, Los Alamos CIS model size version 4. These are some characteristics of the model setup. The resolution that I'm using is, in these experiments, is about 4 kilometers in the North Atlantic, and I have configuration which has about 2 kilometers in this region. And along the Greenland coast, as well as some other locations which are not the topic of today's presentation, the passive tracers are released during the simulation along the coast at the locations taken from the Jonathan Bamberg data. So these are very accurate locations. This is the animation showing the distribution of the passive tracer in the upper layer. So you can see that the passive tracer spreads from the coast of the Greenland. They're released at every location along the coast, and it goes into the North Atlantic. So we can see from this animation that Greenland freshwater generally follows the propagation pathway of the Greenland Great Salinity Anomaly. So with the boundary current around the Greenland along the shelf, and then it spreads into the Labrador Sea and the subpolar North Atlantic. The regions that are mostly strongly affected by the Greenland freshwater anomaly is the Labrador Sea, other Labrador Sea, the SPNA, well, SPNA includes Labrador Sea, and Baffin Bay. And you can see that even after 10 years, the Nordic seas are almost not impacted by the Greenland freshwater anomaly. And this is the main difference between the Greenland freshwater anomaly pathway and the freshwater going from the Arctic Ocean, because this freshwater from the Arctic Ocean stays more on the continental shelf, and it can be more easily advected, laterally advected into the Nordic seas, whereas the Greenland runoff stays very close to the coastline. The main outflow of the Greenland freshwater is over this region, Illabank. By the way, this is the region where the GSA was observed the strongest signal. So it seems that the pathways are quite comparable. All right, so I don't have time, of course, to talk in detail about propagation and mixing of the Greenland freshwater anomaly in the subpolar seas, but I would like to spend a little bit of time on looking at the Greenland freshwater anomaly on the Greenland shelf. So first of all, the question is where the main outflow of the Greenland freshwater from the shelf into the interior basins. And we saw this in the animation that I showed before, but this analysis, which calculates the total Greenland freshwater flux across this contour, which goes around the Greenland, roughly following the continental shelf break, this calculation shows that the largest fraction of the total freshwater flux is across this part of this sector. This is the south western part of the Greenland shelf. It's about 85% of Greenland freshwater leaves the Greenland shelf across this segment. Another interesting point is if you look at the vertical distribution of salinity along this contour. So this diagram shows this salinity distribution along this contour. So I kind of cut it and unfold in this diagram. So the 0 point is here, it's here on the left, and then we go counterclockwise along the contour. And the vertical distribution of salinity shows that the outflow is very well mixed. This is the region where the Greenland freshwater outflows from the shelf. So why is it so? I hypothesized that this is the wind-driven mixing, which is very strong on the Greenland shelf. The Greenland shelf is dominated, the eastern Greenland shelf is dominated by very strong downwelling favorable winds. On the western side of Greenland, the wind is more variable. It is upwelling favorable for winter, and in summer it alternates between downwelling and upwelling favorable. So if we calculate the Ekman transport from the winds, we can see that winds are pushing water onto the shelf on the eastern side and pushes water from the shelf on the western side. It is also interesting that the Greenland freshwater on the shelf responds very well to the wind. Actually, the buoyant plume theory, which has been developed for slightly smaller scales, is very applicable to the Greenland freshwater on the Greenland shelf. These are on the right are the vertical sections showing salinity in colors, and also the black contours show the salinity values from the high resolution model. And in winter, the bottom diagram and the cross section is the snap section on the southeastern part of Greenland shelf. So in winter, when the shelf is dominated by very strong downwelling favorable winds, we see that the isochalines are almost vertically oriented, which means that the winds pushes all fresh water all the way down to the bottom. That's why it's so well mixed. Next question is timescales, because we want to know how long the Greenland freshwater anomaly stays in the subpolar North Atlantic. One theory is that this freshwater doesn't stay too long. It comes in, comes out. That's why we don't see any impact of this increased Greenland runoff onto the salinity in the domain. Another theory is quite opposite, that it stays in the subpolar gyre for a while. It's just getting mixed down and down. So we want to estimate the residence time and the response time of the system to the perturbation of the freshwater flux. So for this, I suggest to use a relatively simple first-order dynamical system, which describes accumulation of the Greenland freshwater anomaly. V is the volume of the Greenland freshwater anomaly inside this contour. We have forcing function, f of t, and which are the fluxes, and we have direct fluxes from the southern Greenland, and estimating from the Jonathan Bamberg data, I found it is about 70 cubic kilometers per year. We also have indirect fluxes of Greenland freshwater with oceanic currents through Denmark and Davis Straits. And these estimates are quite uncertain, I have to admit this, and I use the model simulation to estimate these fluxes. But anyway, I'm getting these numbers, and they do make sense, but as I said, there is quite a bit of uncertainty in these estimates. The important term here is the outflow. So here I hypothesize that the outflow of the Greenland freshwater anomaly is proportional to the amount of the freshwater, Greenland freshwater anomaly, accumulated inside this domain. So this relationship is assumed. And I want to find this coefficient k, which is the inverse of the response timescale, tau. We can easily obtain the steady state solution by applying the step forcing, which is not of interest for us. We're interested actually in the non-steady state solution, because the Greenland freshwater discharge is not steady, it's increasing in time, it's accelerating in time. This Greenland freshwater discharge can be approximated with this linear trend. So we use this forcing function, and this is solution obtained to this problem that I showed in the previous slide. So it has two parts, the linear part and the exponential part, which quickly decays in time in the time scale of the tau. Then I use the estimate from the model, compare with the V from the model, derive the time scale, which is about 13 years. The question is, how valid is our assumption that there is a linear relationship between the outflow and the amount of fresh water accumulated in the domain? So you can see that I'm showing here with red is the amount of fresh water from the model, and also I'm showing the outflow also estimated from the model, and they're very well related. Actually, I found that the coefficient k from this regression is also about 13 years, which agrees very well with the analytical solution. So I think that this assumption is valid. And next, we also interested why the response is so different to the Greenland salinity anomaly. GSA can be viewed as a pulse of fresh water which entered the subpolar North Atlantic and propagated with the currents. So I modify the forcing function with a combination of two Heaviside step functions. One is turn the forcing on, the other turn it off, and I'm getting this solution for the analytical model. This shows the diagram shows the accumulation and release of fresh water when the system is impacted by this bump of fresh water. We see the very rapid increase, and then it slowly decays in time. If we use the estimates of the GSA from published papers, actually I'm using Curie and Morris in 2005, I found that the time scale is about 11 years. It doesn't have to be the same as the Greenland freshwater anomaly, and I'm not certain why they shouldn't be the same, but anyway, it seems to be that according to analytical solution, the time scale for both cases are comparable in the order of a decadal scale. And next, I want to find the residence time scale from Lagrangian experiments, just to compare with the analytical solution. And for this, I do the Lagrangian experiments, release Lagrangian particles at two locations in the northern Labrador shelf and on the southwestern shelf of Greenland, shown with the blue dots and orange dots. I also track propagation of Lagrangian particles at 4 different model layers. This depth are not the release locations. This depth are the layers of HICOM, which is a Pickman model in the deep basin. They all released on the shelf, which has less than 450 meters depth. Keep this in mind. And the particles are directed for 26 years. So I'm not going to show the animation of the particles just for the interest of time. I'm showing the final result, which shows the probability of observing a particle in a grid cell for particles released at the southwest Greenland shelf here, and particles released on the northern Labrador sea shelf for different depth layers. So what we can see the same between the two group of particles is that the particles in deeper layers tend to circulate inside the deeper basin and stay longer in the subpolar North Atlantic. Particles in the upper layer, they actually quickly leave the domain. and they tend to stay on the shelf with the boundary currents. However, there is a big difference in the pathways of the particles released at the Labrador Shelf and southwest Greenland Shelf. The Labrador Shelf particles stay mostly inshore, and they quickly leave the domain. Very little particles enter into the interior subpolar North Atlantic, whereas the Greenland Shelf particles, southwest Greenland Shelf particles, they quickly spread into the interior subpolar North Atlantic. These are the particles age estimate from the Lagrangian analysis. The blue is southwest Greenland Shelf, and the orange is North Labrador Shelf. We see that, first of all, the increase of the particles age with depth. So particles that in the deeper layers stay much longer than the particles in the surface layers. And also the particles released from the Labrador Shelf have shorter age. So they stay shorter period of time. So the next question is how this residence time of particles related to the response time. These are not the same time scales, but then they're related. For example, the shorter residence time means the shorter response time. And there is a whole theory how to relate these two time scales. For the steady state solution, it's very simple because these time scales are the same, but we have non-steady state solution. So I'm not going to go into the detail, into the derivation. So I was able to derive this relationship for our case. And what it shows is that the residence time scale, which are shown here, they asymptote the response time scale as time goes to infinity. In other words, we can estimate the response time scale, and it gives us the upper limit of the residence time scale of the Greenland freshwater anomaly if the process continues for years and years and years. For 6 and 7 years, the residence time scale has not approached yet the limiting case, so it is shorter than the response time scale. And in order to compare this with the analytical solution, we have to keep this in mind, and also we have to do this kind of integration between the particles released from the Labrador Sea shelf and from the Greenland shelf. So to summarize this part, we can see that the Greenland freshwater anomaly has decadal time scale in the subpolar North Atlantic. And the response time scale is mainly determined by the Greenland freshwater anomaly residence time. The quick response of the system is explained by very slow flux of fresh water from Greenland shore onto the shelf. I think that if the flux was much higher, the response would be very different, and probably the system that I used to derive the analytical solution would not be applicable to this case. And also, it seems that most of the Greenland freshwater is accumulated in the subsurface layers, and it stays quite long in the layers below 100 meters. The impact of the, and I'm wrapping up, the impact of the Greenland freshwater anomaly is on the salinity is quite small, because the freshwater is very strongly mixed in the deep ocean. And very small fraction of Greenland freshwater anomaly stays in the upper layers. So this is the colors show the concentration of the Greenland freshwater anomaly from the model simulation. This is the cross section which goes from Baffin Bay around Greenland to Front Strait, and you can see that the vertical distribution is quite substantial. It goes to almost 2,000 meters, although the scale is logarithmic, so there is a big difference between the green colors and blue colors. And the numbers show the centimeters of freshwater anomaly, 50 meters of seawater. I did estimate the salinity change caused by the Greenland freshwater anomaly. It's quite small, very small. It's in the order of 0.02, 0.03 in the central North Atlantic. All right, here is the summary. The Greenland freshwater from the south, traveling from the southwest Greenland shelf, is the main contributor to the Greenland freshwater anomaly that spreads to the interior subpolar North Atlantic. The freshwater, Greenland freshwater, which travels from Baffin Bay, is not that important for the interior subpolar North Atlantic. It quickly leaves the domain. The SPNA responds to increased Greenland freshwater flux not by changing notably salinity, but by accumulating it in the subsurface layers. So if we had very accurate, very detailed measurements of the salinity in the subpolar North Atlantic, we would be, I hope, we would be able to integrate the salinity anomaly in the domain, and probably there we would see the large change of increase of Greenland freshwater anomaly in the domain. And also, the analytical solution shows that the SPNA response to the Greenland freshwater anomaly is different, qualitatively at least, from the response to the GSA type events. The freshwater quickly accumulates during the GSA type events, and that it slowly releases the freshwater, whereas during the Greenland freshwater anomaly, the anomaly is slowly accumulated in the subpolar North Atlantic, and it's still accumulating. So as long as we have accelerating Greenland melt, I guess the Greenland freshwater anomaly would be accumulated in the subpolar North Atlantic. And with this, I'm done. I'm ready to answer the questions. Thank you.
Thanks, Dmitry. So if you have any questions, please use the raise your hand button, or you can type it into the chat box. We'll take the next few minutes for questions.
I have a quick question, Dmitry.
Yes, please.
I don't really understand your second point. You talk about the response of the subpolar North Atlantic gyre, and you say that it's increasing the freshwater flux, it's growing the volume of freshwater anomaly, but it's not changing the salinity. Is that what you're saying?
Well, it's it does change salinity. It does change salinity, but I'm going to find the slide. I'm sure I have it somewhere in the back here. It does change salinity, but salinity changes are very, very small, and they cannot be really measured because these changes they're masked by some other process like precipitation, for example. This is the estimate of the salinity change from the passive tracer numerical experiment, where analyzed how much fresh water is accumulated and what would be the impact for this extra fresh water on salinity in the top 50 meters. And you can see that the interior subpolar North Atlantic probably experiences very tiny salinity change in the order of minus 0.02 PSU, which is very small. For reference, if I remember correctly, during the GSA, salinity change was in the order of 0.4.
And GSA stands for what again?
For Great Salinity Anomaly.
Okay.
Great Salinity.
All right. And these changes, sorry, on the time scale of what? The experiment. I'm just trying to understand the size with respect to the time scale. So.
Sorry, what was the question? This change what?
So these changes that you're showing on this slide, they are taking place on which amount of time on average?
Oh, yes, this is for 2016.
Okay. All right, thanks.
Yeah.
Question from Allison.
Hi, Dmitry. Thanks for the really nice talk. I guess my question is a little bit broad, but I was wondering if you had any thoughts about sort of what, the implications for what you've shown here and your results for like OSNAP and how we're measuring changes in the subpolar North Atlantic. Is that, you think, going to be well captured by like how things are arranged there, or do you have any thoughts in general about observing these changes in the real ocean?
Yes. Well, I'm not an observationalist, and probably Isabella would correct me. I don't think that in the interior subpolar North Atlantic we can really measure the impact of the Greenland freshwater anomaly. I'm pretty sure that the signal should be quite strong on the shelf, but in the interior subpolar North Atlantic, unless we use geotracers, I don't think we can derive any signal from salinity measurements only.
Okay, thanks. Thanks.
Any other questions for Dmitry? There is a comment by Isabella, so if you want to take a look there, she says she agrees the shells are also very difficult to measure with moorings.
Okay.
So I think we'll end the pause webinar here. Stay tuned. I'm not sure if we'll host another webinar in January. It'll fall on January 3rd, so I don't know if it's be too close to the holidays, but I'll send an announcement out once we have that scheduled. And if you're part of the pause panel, please stay on the line as we host the 30-minute tag up right after this. Everyone else, thank you for joining. And a recording of this webinar will be available next week. Have a great day.